Okay, so this is a related talk to the one I gave earlier on the protease detection, and I'm going to show you how all these biomarkers together are able to give you a better idea of what's going on, especially in early cancer. This is this work predates the work that I talked about, the protease work that I talked about in my earlier talk. Okay. And this is a sample to answer electrokinetic device for multi-omic biomarker analysis in cancer patient blood samples. Okay. Now I have a disclosure here because I'm a member of the scientific advisory board. The technology, this electrokinetic technology was developed in my lab at UCSD, along with the protease detection technology. And of course, I mean, pretty much everyone here is well aware of what we're doing here in the cancer area for diagnostics and therapeutics. We have cancers. They're releasing, in the case of diagnosis and early detection, we want to try to find the biomarkers, which can be the cell-free DNA with the mutations. Epigenetic markers could be the RNA, and then it also can be the exosomes, extracellular vesicles that have specific tumor-related protein biomarkers. And of course, having been in this for such a long time, okay, what we realized is there's a lot of just pragmatic reasons as to why it's so hard to do early detection, okay? There's just hundreds, if not thousands, of attempts made at designing technology for this kind of molecular diagnostics. And most of all, 99% of them fail. Generally, it's the sample preparation, it's too time consuming, too labor intensive, complexity, it costs too much. Clinical samples themselves are an issue in that you can get a result with a clean sample spiked with the biomarkers that you're interested in, but you look at a clinical sample, it doesn't appear to be there. And that's reasons why that goes on. We developed an electronic device, a microarray chip that applies electric field. And it's as you see in the little cartoon here, it's able to isolate all of the high molecular weight, large biomarkers. This would include cell-free DNA, nucleosomes, exosomes, everything that shouldn't be in the blood that contains the biomarkers gets pulled down on this chip. And then once it's there, you wash the rest of the blood away, okay? And now you have the cell-free DNA and the biomarkers, the exosomes of interest on the chip. And the company we started is called Biological Dynamics. To make a long story short, the technology is developed at the company. We have lots of patents in this area. And of course, the big thing now is to turn it into viable diagnostics. They have a number of devices that they've developed, including clinical lab, research devices, and even a little point of care system, a handheld system, just like the little glucose sensors that you buy at the drugstore. You know, you buy it for, $50 or something and put the blood on it. So why isn't that being used for, if the technology is there to do this kind of thing, then why don't we have a little sensor like that out in your drugstore? The reason is because the biomarkers, unlike the glucose and biomarker for diabetes, and so the biomarkers for cancer have not been validated and correlated with the disease. So in some cases, in some aspects of the technology are way advanced over the ability to translate it for patient use or home use. And we even have movies. You can take a blood sample from your finger, put it on this thing, and you get a result in about 10 minutes. So that's an interesting caveat about technology and how aspects of technology can be advanced, so advanced, and yet it doesn't reach the market. These are just some pictures of results that you can get off of that little device that we've seen. You're able to measure the cell-free circulating DNA in there. In this case, we're looking at one of the biomarkers. This is for lung cancer, PD-L1. You can pick these things up. We've published many papers. Of course, we're in academics and we have to publish papers, and we get lots of nice papers and everything. And yet still, technology takes a long time to reach the patient. This is just a little more background on the devices themselves. These are what are called a dielectrophoretic microarray. There's several hundred of these little platinum electrodes on a little microchip, maybe a centimeter in size, and you can actually watch if you stain the exosomes or the DNA, you can actually watch it go down on the electrodes, and then later, as you see here, you could see here's the little exosomes where we did SEM images, and they're sitting on there. And then you can do all of the downstream analysis. You could take the DNA, do sequencing on it. You could take the exosomes, do immunofluorescence analysis of the biomarkers, the protein biomarkers on those exosomes. We've been able to do lots of different things. We've done CLL mutation analysis in the DNA from CLL patients. We've published on that. This is just a little, schematic of the process. You take a whole blood sample, plasma, serum, put it down on the little chip, turn on the electric field, pull everything down, add an antibody, wash it, do the on-chip analysis, fluorescent analysis, then take the DNA or RNA off and do further genomic analysis with that. And just a little bit of what things actually would look like stained on the chip here. Here you see CD63, glypican-1, and then just a bright field image of what the chip is. Here's the little electrodes here. And again, we've done a pancreatic cancer study. Our earlier work, we did a lot of work on pancreatic cancer, and we did looked at the glypican-1 levels in PDAC and in colon cancer. And we've been able to, these are very small samples, 10 to 20 microliters blood plasma or serum, get relatively good results for the level of these biomarkers. The caveat being that biomarkers like glypican-1, exosomal biomarker like that, are relatively specific. They're not absolutely specific for pancreatic cancer versus colon cancer. So what one realizes is that if you want to really do these diagnostic assays, you need to go to multi-omic, where you look at more than one biomarker. You might want to combine genomic biomarker with the exosomal biomarker. And that's what we were able to do. These are just some of the results. These papers are all published. Anyone that's interested in this, you can send me an email, mheller@ucsd.edu. I'll send you anything that you want. Just for the sake of time, I'm going to go through all of the work that we did because I'm trying to make a final point to all the cancer researchers. This is other things that we did. This is brain cancer. We were able to pick up same thing. We could find exosomal proteins that were leaking from the brain with people who had glioblastoma, and we've published on that. Again, very small amounts of sample. And interestingly enough, we did a further study with traumatic brain injury. Okay, and I'm sorry, I'm going through the further analysis of the brain proteins for brain cancer patients in normal plasma serum. So we see the results from glioblastoma patients. And then we now did a big study for the military where we looked at biomarkers for mild and moderate TBI, okay? And we looked at a whole bunch of samples, and these people, not severe TBI, but people would come into the emergency room, fall down, bump their head, they'd get a CT scan, and 9 out of 10 times they were okay, but maybe 1 out of 10 times they would go on to have severe problems and further brain damage. And that would be picked up by the CT scan. So the goal was to develop a blood test to see if we could pick up the brain proteins in a small blood sample. And again, to make a long story short, we were able to do that. We were to look at several dozen samples, most of which did not have positive CT scan. And then the ones that did, we got perfect correlation with the where we saw the leakage of the brain proteins for the traumatic brain injury patients. Okay. And we also did demonstrated the cell-free DNA and exosomal biomarkers for prostate cancer and integrin. We were able to do that. The other thing that we noticed is that there is a high level of what we would call high molecular weight cell-free DNA that you find in cancer patients, okay? So those of you who are aware of what's going on in the genomics area, right now, most of the next generation sequencing is based on looking at mutations in the small apoptotic DNA fragments that are released from the tumor. But there's also this large population of high molecular weight DNA, which we've determined also has the mutations in it. And so again, that little thing that I've shown you, we are able to take a sample, put it on the chip, do a relative level of the cell-free DNA, high molecular weight cell-free DNA, do immunohistochemical staining for the protein biomarkers on the exosome, and then take the DNA off, which is the high molecular weight DNA from the tumor, extract it, carried out digital PCR, and then sequencing of that. And so I don't know how many people are familiar with digital PCR, but it's a very powerful technique for looking for mutations in very low copy numbers of DNA. So we carried all of this out to make a long story short. We did the cell-free DNA digital PCR work. We did sequencing. And then we were able to see when we did Sanger sequencing, mutations were in that high molecular weight DNA that we had identified mutations by digital PCR, the same mutations by digital PCR. We put that whole study together. It's a very complex thing here, but just bear with me for a minute. So what did we see? We had 36 blinded samples, 12 of which were benign, 12 of which were various stages of pancreatic cancer, pre-PDAC cancer, and we had 12 that were different stages of PDAC, okay? So what you saw was relatively the level of cell-free DNA increased as you went to the cancer patients. Relatively, the level of glypican-1 exosomal biomarker increased as you went to the more severe pancreatic patient samples, okay? Relatively, the number of mutations that you saw in the digital PCR and the Sanger sequencing increased as you went to the cancer patient samples. And I'm stressing that word relatively, okay? Because any one of those things by itself, any one of the genomic or the proteomic biomarkers by itself was not good enough to make the call on these samples, to differentiate by benign from pre-malignancy versus the actual pancreatic cancer patients. Okay? So we put that together in a bivariate analysis. Okay, we had gotten most of the patients right, but we had two stage 2 cancers down here that you can see here. Okay, and they were discordant. We later went back and looked at those same samples with several more of the exosome protein biomarkers, and they now moved, we were able to move them into to make positive calls on them. It was kind of after the fact, but the goal is to figure out when you see something that's discordant, why is it discordant, and how do you eliminate discordancies when you're doing these analyses. So now the thing is, of course, multi-omics, which is the direction that many people are going in, it's much more time-consuming, much more complex, but that seems to be required to make a reliable positive call, and also to start differentiating which stage the cancer is. Okay. Now I want to go back to my first talk, okay, which I think some of you heard, where I showed the protease results. We were looking at the proteases that were released in pancreatic cancer. After that genomic study, multi-omic study was done, I took those same exact samples and I started running the proteases assays for chymotrypsin, trypsin, elastase, and then for the MMPs and the cathepsins and kallikreins. So what did I see? Okay, sure enough, okay, for most of the cancers, most of the PDAC cancers that we picked up with the multi-omic approach, okay, we also saw the elevated levels of the protease biomarkers. And for those markers that were elevated, the ones that I would call the hottest samples that came up by cell-free DNA level, by glypican-1 or the other biomarkers, or by the KRAS mutations. Now, when we looked at the proteases, they had the highest levels of elevated proteases, and they were the ones that had also caused the elevation of the secondary set of proteases, the MMPs. So again, as a person who's been in this a long time, what I'm trying to convey is how complex this whole thing is, and how there's so many things going on in this disease that in order to get a full picture of it, you have to look at least, probably, I'm guessing, a half dozen of these biomarkers. So what do we really need for early detection? We can't afford to be screening people several times a year, taking 10, 20 mLs of blood at a time, huge expense to pick up these early cancers. We need what we would call a fire alarm, okay? Something that basically tells us in a simple, fast, easy way that there's pathology there. Once we know that, then we can take that particular group of people that we've screened in a simple, fast, easy way, and now we can do the more expensive genome testing, next generation sequencing, and things like that to determine exactly what's going on. This is, as a person who's been in this a long time, this is just the way I think everything is going to turn out, and we'll see if it does. So again, remember that in this area, early detection is important, because for cancer, the earlier we can detect it, the better the outcomes, and most of the people in the audience are well aware of that. We've published a lot in this area. Anyone that's interested, please just send me a email and I'll send you whatever you would like. Again, I'm very lucky. I get to work with lots of good students, postdocs, and clinical people, so I can, present things like this. There's still a lot of work to be done out there, and a lot of discovery, but I think probably we're far along on our quest to do early cancer diagnostics. And I think now a lot of the effort is just simply in order to make it viable, or to translate into usable technology, to bring the cost down and the ease of use. So again, thank you for letting me give a second talk here, and hopefully if anyone has any questions, I'll be able to answer them. Thank you.
Thank you, doctor. Any questions from audience?
Okay, was that good? No one has any questions?